Hi, welcome to my presentation. Today I'll be walking you through a presentation or a telling of a story of sorts about a fish that has been found to be able to survive and thrive deep within the Mariana Trench. This fish is the Pseudoliparis swirei. I hope I got that right, or colloquially termed the Mariana Hadal snailfish. So specifically, I'll be explaining the features and changes found on these fish and how these changes help them survive well under extreme conditions. So let's start off with a brief description of the ocean. So this diagram in black and white, it shows the relation of biomass, light penetration, and temperature to depth in oceans. The temperature structure of oceans is affected hugely by the alteration of density of seawater with temperature. Warm seawater is less dense than cool seawater, and so warm water tends to lie above cooler water. So a stable system is typically formed in tropical latitudes where a clearly defined layer builds up, separated from the underlying water by a sharp temperature boundary called the thermocline. Thermoclines also form in temperature latitudes in the summer, but when surface water cools down in the winter, the system loses stability and the layers will mix. Below the thermocline, the temperature falls slowly but steadily with depth at 5 degrees Celsius at 100 meters to about 2 degrees Celsius below 4,000 meters. Pressure in the sea rises 1 atmosphere for each 10 meters in depth. So deep sea fish are commonly exposed to pressures of some hundreds of atmospheres. All life in the ocean is ultimately dependent on light and the ability of the ocean flora to photosynthesize. The euphotic zone is where all the primary production is concentrated, where many of these small planktonic animals are found. Short and long wavelengths of light are rapidly diminished with depth, leaving only blue and green light that penetrates the deeper levels. And below the euphotic zone is called the twilight zone, where intensity of light decreases and the spectral band narrows. So as you can probably imagine, it is very, very dark down there. But really, we're mainly focused on a zone called the hadopelagic zone, which lies deep within the ocean trenches. Overall, the hadal zone is the deepest region of the ocean, and it represents 45% of the ocean's depth range. It is also one of the Earth's least explored habitats. And a fun fact here is that it was actually named after the realm of Hades, which is the underworld in Greek mythology, which actually seems pretty fitting, I would say. Its depths range from 6,000 to 11,000 meters, and they are among the most hostile environments on Earth due to their high hydrostatic pressure, darkness, limited food resources, low temperatures, and hypoxia. The most conspicuous environmental constraint in the hadal zone is hydrostatic pressure, which increases by 10 atmospheres per 100 meters of depth, reaching about 1,000 atmospheres in the deepest ocean trenches. Most of the hadal zone is made up of subducting trenches located around the Pacific Rim, and they exist in long but narrow topographic V-shaped depressions, as shown here in the figure to the right. The common creatures found in the hadal zone of the ocean includes amphipods, isopods, decapods, sea anemones, nematodes, and cuscales, and most of these trench communities probably came from the abyssal plains. Typical midwater deep-sea fish, as we know, include the anglerfish, viperfish, bristlemouth, and rattails. These fish are known for their sharp teeth and rather scary looks, which is why the discovery of the snailfish is so fascinating, to me at least, because the snailfish is rather small, it's pink, and one can even argue it looks rather cute. So now let's transition into a section where the hadal snailfish was discovered and described. So basically, a researcher from the University of Washington played a key role in discovering this new species. The species was actually first caught in 2014 and again in early 2017, but was only formally described and documented in 2017, which is this paper right here. Essentially, they fished out the snailfish at about 8,000 meters or 26,000 feet in the Mariana Trench, and these fish are thought to be able to handle pressures equivalent to the weight of 1,600 elephants. The researchers used free-falling traps baited with bags of mackerel to catch and retrieve the fish. They also recorded footage of the Mariana snailfish swimming at extreme depths. So since they did record the footage of the fish, let's enjoy this short clip of the fish eating and enjoying a meal and swimming around. I hope all of you like that. So now let's go into details of what the snailfish actually look like. Well, essentially, the Mariana Hadal snailfish are translucent, flabby, and gooey creatures. They belong in the Scopaniformis order and in the Leparidae family. A fun fact here is that the name of the species was actually given after a navigator on the 19th century expedition that discovered the Mariana Trench as a form of tribute. These fish also have the widest depth range of any marine fish family, with habitats ranging from the intertidal to depths exceeding 8,000 meters. Here I will be describing, based on a paper published in 2017, which is the blurb that we all saw earlier, the Mariana Hadal snailfish. And essentially, they have a small head with a low and wide profile, which is anteriorly rounded. They have a blunt snout and a broad subterminal mouth. Their eyes are small, at about 10% of their head length. Their upper and lower lobes are also closely spaced. They have a small gill opening located completely above the pectoral fin. Their pharyngeal teeth are well developed, long and sharp, and overall, they have a pinkish white body, and their internal organs, such as the liver and the stomach, are visible through the skin and the thin abdominal wall. It's pretty amazing how these snailfish is able to survive deep within the Mariana Trench, but how are they actually able to do that? So here I'll be attempting to answer this question by going over a few features and characteristics of the snailfish that help it thrive and survive in an extremely stressful environment. Based on advances in deep diving and genome sequencing technologies, a study done in 2019 revealed pretty interesting insights about the morphology and the genome of the Mariana snailfish that really help it thrive and survive in the cold, dark, and high-pressure environment of the Hadal zone. The first here is that the bone GLA protein has a frameshift mutation that may cause early termination of cartilage calcification. Vertebrates living on the surface of the earth have closed skull spaces surrounded by hard bone to protect the brain and to maintain appropriate intracranial pressure. But closed skulls cannot maintain their structural integrity under very high pressures of the Hadal environment, which makes it really necessary to have an open system. So what they found here is that the skull of the Mariana snailfish is not completely closed, allowing for internal and external pressure equalization. Most of their bones also consist of cartilage instead of being ossified. So the premature termination of the bone protein in the Mariana snailfish may be associated with the species' unusual skull structure and reduced bone hardness. Previous studies have suggested that mutation rates are sensitive to many factors, including environmental energy, metabolic rate, life history traits, and especially generation times. Hadal species reportedly have comparatively low metabolic rates, so the Mariana snailfish may have a slow life. Coincidentally, the researchers also observed that the female Mariana snailfish made fewer but larger eggs than females of other snailfish species, suggesting that they may have a specialized reproduction strategy to increase generation times. And these specialized reproduction strategies can include eggs that hatch at juveniles rather than at the larvae stage. Apart from bones with reduced hardness, there are also changes to the cell membrane of the snailfish. The cell membrane is a lipid bilayer containing different proteins. High hydrostatic pressures reduce the fluidity of lipid bilayers and the reversibility of their phase transitions, which leads to the denaturation and functional disorder of membrane-associated proteins. Pressure also rigidifies the membranes, which impairs their transport functions. And this group of researchers found many significantly expanded gene families in the Mariana snailfish, and the gene families exhibiting the most significant expansion were those related to fatty acid metabolism. Phospholipids are major constituents of cellular membranes, and their fatty acid composition is regulated to maintain membrane order and fluidity. Other biochemical studies have also suggested that the membranes of deep-sea adapted organisms contain a higher weight percentage of unsaturated fatty acids than any other organisms living in shallow parts of the sea. The researchers found 15 copies of a gene called ACAA1 in the snailfish genome, while other fully sequenced teleosts only have 5 copies. This gene encodes the acetyl-CoA acetyltransferase protein in the biosynthesis of DHA, which works to alter basic properties of membranes such as fluidity, elastic compressibility, and protein activity at high pressures. They also found increased copies of another gene called FASN, and together these changes may increase the abundance of fluid membrane lipids, which enables the survival in the world's deepest ocean trench. Other significantly expanded genes include the TFA and SLC29A3 genes, which belong to families with ion and solute transport-related functions. This is consistent with the need to resist high pressures, which induce inhibition of fluid transport in hadal organisms. Other positively selected genes, which is a process by which new advantageous genetic variants sweep through a population, also exhibited functional enrichment with respect to transmembrane support, ATP binding, and ion transport. Hydrostatic pressure strongly constrains protein function, which affects both folding and enzyme activity. So, species living at great depths must maintain an intracellular environment that preserves the intrinsic properties of their proteins. One way this is done is through the physiological adaptation mechanism involving accumulating small organic solutes such as trimethylamine N-oxide, or TMAO, to preserve protein at elevated hydrostatic pressures. TMAO is a physiologically important protein stabilizer that can restore denatured proteins to their native structures. Its abundance in teleosts increases with depth, and deep-caught species have significantly higher TMAO levels in all tissues than shallow species. Most teleost genomes contain 5 copies of the TMAO-generating enzyme flavin monooxygenase 3, or FMO3, 4 of which are tandem repeats. The first gene of these repeats, called FMO3A, was strongly expressed in the liver of the Mariana snailfish. These changes in the gene's protein coding and regulatory sequences may really help the snailfish increase TMAO levels to improve its protein stability. Structural adaptations of proteins to deep-sea conditions may include changes in amino acid substitution patterns and protein structure that counteract the effects of pressure on protein function. The only gene family found to exhibit convergent amino acid changes was a gene called HSP90. HSP90 is an evolutionarily conserved and highly abundant molecular chaperone that promotes the correct folding and activation of over 200 proteins. And these proteins are involved in essential cellular processes such as signal transduction, cell survival, and responses to cellular stress. Therefore, the recurrence and the fixation of the substitution in such a conservative site indicates that it is very likely to be beneficial for the adaptation of the Mariana snailfish. The environment 7,000 meters under the sea is also completely devoid of any light. Genomic analyses also reveal that it has lost several important photoreceptor genes. First, they found that rhodopsin, which is encoded by RHO and regenerated by RGR, these are both genes, is an extremely light-sensitive receptor protein found in rod cells that is responsible for low light vision. The Mariana snailfish has also lost its skin pigmentation and become transparent because the most well-known pigmentation gene, called MC1R, has been completely lost in this species. Another characteristic of the snailfish is that it is a selective predator. Analyses done by another researcher found that amphipods were the most abundant prey item in the stomach content, and its abundance increased with depth in the hadal zone. They also have highly developed and strongly muscularized pharyngeal jaws to handle and process large live prey. It's posited here that trophic interactions may be important evolutionary drivers of depth zonation patterns in abyssal and hadal fishes. So, on the upper edges of the trench, the hadal fish community resembles the community found in the abyssal plains. However, from depths ranging from 6,500 to 8,200 meters and onwards, in a number of trenches, the fish fauna seems to shift to a dominance by the Leparidae family. So, while scavenging fishes do not extend far into the hadal zone, suction-feeding predatory fishes are pretty dominant. The increased amphipod biomass in the hadal zone compared to the abyss may provide little benefit for other types of fish and organisms to descend further down into the hadal depths, but it poses a large advantage for suction-feeding fishes such as the leparids. This may be one of the reasons why the Mariana snailfish and overall leparids, they are so notably successful in many hadal trenches. There are also other adaptations known in the leparid family for them to survive in extreme depths of the ocean. For example, some Antarctic species have antifreeze protein in their skin. Others have dermal ossicles that may offer protection with minimum additional weight. And without the swim bladder, some of them actually achieve neutral buoyancy through the decreased skeletal ossification. And some of them also have a subdermal extracellular matrix. And that is the end of my little presentation. I just want to say thank you for tuning in. It was pretty fun for me to make this and to learn about this species in particular. I hope you've learned something new today. And just for jokes, when you think you're living under extreme pressure, know that these snailfish are under way more pressure than what we can all imagine. As a joke.